Namaskar and a very warm welcome to our week 75 Know Your Species talk, which is being celebrated as part of the Azadi Ka Amrit Mahotsav campaign. So, as you all know, the Central Zoo Authority is taking this campaign forward through a massive outreach campaign titled Conservation to Coexistence, The People Connect. And under the helm of this campaign, we have successfully showcased 75 conservation priority species across 75 zoos in India. This being the 75th week of the celebration, we brought to you three different talks on priority species that are identified in India, that are native to India and are identified for conservation. So today's talk is going to be done by Dr. Karthik Vasudevan, who is the chief scientist at the CSIR Center for Cellular and Molecular Biology, Hyderabad. So Dr. Karthik has done his doctorate on the amphibian species assemblages of the wet evergreen forests of southern western Ghats of India and the effect of forest fragmentation on the diversity therein. He has done his postdoctorate from the Colorado State University and has worked as a scientist for over a decade at the Wildlife Institute of India before joining his present role at this CSIR CCMB Institute. And he will speak to us today more on the Himalayan newt and the other lesser known amphibian species. So over to you, sir.
Thank you, Arundhati. Thank you for making this slide presentation possible. I'm sorry for the glitches, but I think we are now good to go. First, I would like to start off with the announcement that you had made. You had put a picture of a salamander, and you had put there as the Himalayan salamander, but as Tylototriton verrucosus. Now, that is the scientific name that was given to the species earlier, and it has further been refined. And since 2015, we know it as a species which is endemic to the eastern Himalayan region. So this species has no overlap with other populations in China and Myanmar. So this was a very recent development. So in fact, the enclosures in Darjeeling, Padmaja Naidu Zoological Park, we had requested them to change the names of the species on the boards and all that. So that kind of defines how much we know about these organisms. And when we do work on them, we are actually working on the blind because we don't know their distribution, we don't know their natural history, and we build upon very scanty information. So the title I've been asked to speak upon today is on the Himalayan newt and the lesser known amphibian species. So that's a very broad title, but I will try to do justice given the time I have. Now, some people call it as Himalayan newt, some people call it as salamanders, some people call it as crocodile salamanders. So all those are names of the same species, which is Tylototriton himalayanus. And as I mentioned to you earlier, only as recently as 2015, the species was named. Till then, we thought that we shared a species from, which is also found in China and Myanmar. So, with this background, can you go to the next slide, please? Yeah. So, the value of amphibians in ecosystems are not well understood. We name a few species which are supposedly very important for the ecosystems, and indeed they are. But when it comes to vertebrates, small vertebrates like reptiles, amphibians, we really don't know the value of them in the ecosystem. So it's very important to first unravel those, put it out to the public that they play a very important role. Now, in this diagrammatic representation, I have just shown how the nitrogen moves from plants to insects, and then insects could just die out in the soil, and then the nitrogen is taken up back to the trees to grow, or the plants to grow, and some escapes in the form of ammonia. But what frogs do is actually voraciously feed on insects and convert it into an excretory uh, byproduct, which is called as urea. And all of us know urea actually rings a bell because urea is a very important input in agriculture. So in the second half on the right hand side of the slide, you can see how frogs uh, play a role in transferring nitrogen back into the soil very effectively and also consuming the phytophagous insects that in turn become pests of our crops. So they play a very vital role in this ecosystem. Now, this is not just a cartoon, uh, a slide. I will just request you to move to the next slide where we have actual facts, and they do excrete a significant amount of urea. We have measured it with different frogs, and they play a very important role in the ecosystem. And it seems that now people are developing ways to encapsulate urea into plastic beads and then slowly release urea. And this actually the frogs are doing. So the modern technology which is being developed in China and many parts of the US are to develop slow release urea technologies, and which frogs have been doing all through their life. So uh, we have a very important service that is rendered by these animals, which we need to recognize before we look into their status and understand how we can play a role to help them not decline or even improve their populations. Next slide, please. So a recent evaluation, this happened in 2021, a global amphibian assessment. This happened periodically, but it didn't happen over almost 10 years or more for India and uh, neighboring countries. But it took place in online mode over a period of almost 8 months, and a very extensive participation of people. More than 120 people participated in this evaluation. This is a very busy slide, but I would like your attention to the highlighted cells. On the left, you have the non-endemics, and the right you have the endemics. Now, GA1 represents the data that was collated in 2002 or 2003, and then you have GA2, which is in 2021. And you see pretty dramatic shifts. First, we'll look at the non-endemics. I mean, non-endemics means they don't necessarily have their entire range within India. So very few of them, um, you know, uh, basically less than 10 are under the threatened categories, which is a good thing because we don't have a big burden of rescuing many species that are shared with neighboring countries. But you look at the endemic status. Now, when you say endemic, that means the species which are found only in India and nowhere else in the world. That becomes entirely our responsibility. What we do with them will dictate how they continue to survive in the wild. Now, a significant rise in the number of um, endangered species in amphibians from the earlier assessment. Now, this could be because many species were not assessed in the previous assessment, they were included in this. But this list, about 157 species that require conservation attention. How many are in the zoos? How many are listed in the Wildlife Protection Act? This is of a grave concern because in the previous slide, I highlighted the value of amphibians. Now, how we translate that value to action is the question. And that's where all my further slides will dwell upon. So we really need to understand that we have a very big responsibility. 157 species of amphibians requiring conservation attention by different means, maybe in situ, maybe ex situ, but we need to pay attention to them. That is where we stand. Next slide, please. So, coming back to the Himalayan salamander, I wanted to show you that this species was identified not just based on morphology, because you find that all the uh, Asian salamanders, particularly the group belonging to the genus Tylototriton, have a very similar morphology and very difficult to distinguish. So we used um, DNA-based distinction of the species groups, and it turns out that these species have probably radiated from the Asian Southeast Asia to Eastern Himalayas. So the species that you find in the Eastern Himalayas is an offshoot of what has happened, rapid diversification that has happened in the Eastern Southeastern Asia. So in that paper, the authors, I'm also one of the authors in that paper, we said that due to the recent global environment changes and anthropogenic disturbances, the rapid disappearance of the Himalayan wetlands, which is a major habitat of Tylototriton, has probably led to sharp declines in the population of this group. So this is an indication that the species requires conservation attention. Therefore, the ex situ conservation breeding program that is in place, or the coordinated conservation breeding program that Caesar Day has supported, needs to be further reinforced, strengthened with better protocols, better management methods, which will improve both the ex situ population and then also reintroduce them into habitats which originally had the species. So we'll see the threats to the uh, wild population in the coming slides. Thank you. Yeah. Next slide, please. So when we were faced with the problem, this is way back in 2012, 2011, when we started this whole survey and wanted to understand what is happening with this salamander. It was like a riddle, very sparse information, uh, all dating back to a century old. People have gone, explored different areas, but they have deposited specimens in different museums and with no proper locality records. And even if they are locality records, not very clear identity of the exact location. So, very difficult to piece together information. And we struggled with this sort of a background about the Himalayan salamander. Can you go to the next slide, please? So we first, what we did was we put together all the locations of all the known species of Tylototriton and a related genus called Yautrit. We just put it on a map to just see where are all these locations. And then if you can click once, yeah, so you find that the distinction of these genus are very clear. Eastwards, you find Yautritton, and on the western side, you have the genus Tylototriton. Now, what is there within the territory of India is of interest to us, and therefore we went on to explore further by making a geographic niche model of the distribution of the species. Next slide, please. So the focal areas where we knew that there were records of the species and we wanted to identify the populations there. We visited areas in Darjeeling as well as in Manipur, and many locations were collated during this survey. Next slide, please. So the important information that we have already is that the distribution of the species really has a lower limit. I mean, it can't go below that. In 1,000 meters is where most of the distribution ends in elevation, and it goes up to 2,800 meters. The breeding period is also well set, well determined by the monsoons, which is from May to September. They breed in small vernal pools. There is hibernation during winter because these elevations get cold. So they overwinter, and then the larvae actually spend their time in the water throughout the year, and then the next year they come out. So uh, and eggs are transparent eggs, as you can see in the picture, and they are very vulnerable. They have fortunately they hatch very fast, so within 1 or 2 days you will have the eggs hatching, and then the free swimming tadpole survives in the water, and they have, as I just mentioned to you, a very long period of development. Then they come out as small uh, Himalayan salamanders, as you can see here, juvenile, almost or less than the size of a matchstick. So this is the phase which is most vulnerable, and they tend to disperse. They move very far and away from where they have bred. And during this period, they can be there can be events of predation which remove them, or even getting overrun by traffic on the roads, or any other structure barriers which prevent them from moving. And if they are exposed to sunlight and drying up, they will desiccate very fast and die. Next slide, please. So we performed this ecological niche model, and we used locations that we had and confirmed locations from historical records. And then we plotted it on the map, and we generated a possible distribution of the species. And we also compressed the area to which we need to extrapolate the distribution based on the altitude range that we arrived at, which is between 1,000 and 2,800 meters. Next slide, please. So this was the map that we got. The green color area is the total area under which we wanted to extrapolate the distribution of the species. So we have populations of the species, not just in the eastern Himalayas, also in the Indo-Myanmar, Indo-Malayan region, which extends down into Myanmar. So there are populations, good, very suitable areas which can be used by the species which are found there, and also in the eastern part of Arunachal. So that is another probably a distinct population. So that might have relationship with some of the Chinese populations. So our feeling is that we have more than one species of Tylototriton in India. So before we think of assessing the threats and performing interventions, we need to find out how different these populations are, use scientific methods to arrive at it, and delimit their populations, delimit the species identities. That is the first step to do. And we have been successful with one such population in the eastern Himalayas, which is now described as Tylototriton himalayanus. Next slide, please. So these are the images of the ex situ and ex situ conservation efforts, and also an image of the habitat of the species. They typically survive in small to medium, even large wetlands, which are free of predatory fish and excessive pollution, and they are, I would say, pretty hardy species because they are able to withstand a lot of a huge range in temperature, also huge range in the changes in the hydro period, and they are able to survive in these places pretty well. And the conservation breeding program has been pretty successful because it has been able to maintain the population for a very long time now and successfully breed individuals as well. So the the breeding itself is happens in a separate enclosure. This is a display enclosure which is now visible to you. Um, so the breeding in captivity requires certain stimuli like rain, and that needs to be provided to the captive animals. Otherwise, they will not perform the breeding behavior. And though if it is an outside enclosure, the rain that happens outside might have an influence on the behavior of these animals, but it will require certain interventions to ensure that they breed properly. Once they breed, the eggs, after the hatching of the eggs, the tadpoles need to be taken out, kept in captivity, reared, and then brought into a neonatal area and then raised so that they can grow into adults. Can you go to the next slide, please? So, salamanders, fortunately, because of some really good work that has happened from young people in the country, there is a highlight of these animals, like the work done in 2021, where a group of young people found that these animals actually vocalize. They can make sounds, which is really amazing. That we never thought that the salamanders make sounds, but there you have a new knowledge about these animals, that they make sounds. And they have made a nice documentary of the breeding behavior of the species, and much needs to be done to highlight these to the people, and as well inform the ex situ community to highlight this in their interpretation of facilities that house these animals. And then the right side, there is a news item that I was fortunate to receive from another young biologist today, and she says that this has come up in a local magazine, and a lake in Manipur in Senapati district. The locals have highlighted the populations of the salamander, and they say that they are very important um, for their area, and they have said how important these wetlands are, and they have recognized the importance of this species. Next slide. So when we are talking about amphibians in general, and salamanders also included under amphibians, they have identified two diseases, two fungal diseases. One is BD. BD infects all anurans or frogs and toads, and BS, which is another fungal pathogen which infects salamanders. And these two pathogens are global pandemics, and they have been traveling the globe because of the trade with amphibians. Earlier it was for frog leg trade, now it is for pet trade. And this has resulted in a major havoc in causing wild populations to get infected and die out rapidly. And this has become a huge global problem. And in 2014, we started some work on identifying how this disease can be diagnosed for amphibians in India. And we made some progress in that, which I wanted to highlight. So globally, about 500 species of amphibians are infected by the fungus, both frogs and salamanders. And this is supposedly the one pathogen that has caused more extinctions than any other pathogen that we know of so far in the world. 90 species worldwide have gone extinct because of this pathogen. So it's a very serious pathogen, and it needs global attention and globally coordinated efforts. Next slide, please. So what does this pathogen do? So it grows on the skin of the frog. So here in this slide, you can see a cross section of the skin of an infected frog. From the top to down is basically from outside to inside the body of the animal, and the arrows are indicating where the tissue damage. So as soon as the zoospores set on the skin of the frog, they drop hyphae or tubes, and they go inside and they multiply, they grow, and then they form sporangium, or they grow in numbers. And then once the sporangium grows, they will again put another tube going out of the skin of the animal and release all the zoospores. So in the environment, when an infected animal is there, other animals can also get infected because they will release many zoospores, thousands of zoospores, and they will go on to infect other frogs. So a way to detect the fungus early and the way to detect the loads of the pathogen is key to managing the disease. And you can imagine in the wild, if a disease spreads, it's almost impossible to control. It's like fire. In the forest, if there is fire, very little you can do. You can try to manage, but literally you can't do much because the devastation is so fast and so vast. So we have to step in early and be able to intervene at the right time, and so that the results are tangible. Can you go to the next slide, please? So before the slide moves, I just wanted to tell you that the frog actually dies because of a failure of its heart, which is caused by a change in the electrolyte composition in the blood. Basically, it has a heart attack and dies, and within 24 hours of infection, the animal can die. Thank you. Yeah, next slide. So, why this disease? How did this disease come up? And there are different explanations. So, people initially thought that this disease was spreading because the African clawed frog, which is used in the labs to standardize pregnancy testing kits for humans, was moved from lab to lab, and therefore, the fungus also spread. The other alternative explanation was there was trade of frog legs, because frog legs was a delicacy in many parts of Europe and North America. And even today it is in some places. And the supply for this consumption came from Asia. And Asian frogs were collected. In fact, even India was part of the frog leg trade till 1985, when Dr. Humayun from the Bombay Natural History Society intervened and pleaded to the government that it should be banned. And then the Wildlife Protection Act was amended, and freshwater frogs were included in the Wildlife Protection Act. And then there was a ban of trade of frog legs. And in that report, he amply highlighted the value of frogs. So I think following from that, we need to understand that steps need to be taken to highlight the value of these animals in the ecosystem. And can you give one click, please? Yeah. So basically, the hypothesis that is now being trained is the one that that says that the Asian frogs that were transported for the frog leg trade caused the spread of the disease. Next slide. So when we started this work in 2014, this was the map that we were constantly looking at it very worried, because almost all around the world, people were knowing about the disease, what is happening, and how to detect it. But in all these maps, nowhere India was to be seen, and we had no idea of what was happening in India. So we struggled, and we got many samples from all over the country and tried to analyze it and find out what is happening here. Next slide, please. So we could, over 1,800 samples were collected from different parts of India. We assembled it, and then we looked at it. Then we found that the prevalence was very low, around 8 to 12% only of the population, which was sort of a report which matched with what others have been talking about for other parts of Asia. But the number of haplotypes, that is, the variants in that BD, was very high. So that prompted us to investigate further, and we found that some variants are pretty common throughout India, but many of them are new, and we could not match them with any of the global uh, variants that have been reported. I'm very happy that I'm able to use words like variants, haplotypes, because of the COVID, the knowledge that we have acquired through COVID infections. Because when 2013, when we used the word pandemic, it was very difficult to even explain what a pandemic was. So I think people will understand these terms easier today than if it was many years ago. Thank you. Next slide, please. So, going by this, the issue of low prevalence, we wanted to investigate it further. And when we looked at the sequences of the PD pathogen that we got as in our samples, there were so many mutations. Like I told you, so many variants are there. So these mutations will not allow the primers to bind. So the primers are very specific to the sequence of the DNA on the target genome. So if the sequences are altered, then the primers will not bind. So we found such mutations. So the so-called gold standard of detecting BD had to be changed for detecting the same pathogen in India. So we worked on it. So we now have a very good marker which reliably amplifies all the global strains, and it is now a universal marker, and we have used that to understand what is happening in the country. Next slide, please. So we also attempted culturing of the pathogen. This pathogen is not easy to culture. So we did get a culture, but we lost it. It got overrun by another fungi, but we are still working on it. And the reason why we need to have these cultures, just like the way we did, we did find a solution for COVID, we need to have the virus cultured. If you have the virus cultured, you can perform many studies on them, which will result in possible solutions to eradicate the pathogen. And that is what happened in terms of the vaccine development that India was particularly good at. So when we are dealing with a pathogen, the step to deal with them is to first detect them accurately and effectively, and then be also able to find solutions to eradicate them. Next slide, please. So we have been providing these services to different zoos, and we have been able to show them that this new primer that we have developed works very well, and we have been able to detect the pathogen in many frogs, including salamanders. This is BD in salamander, which we never expected, because salamanders are supposed to get BS, and we didn't get BS for salamanders in Padmaja Naidu Zoological Park, but we got BD, which is a good news in one sense that they don't have the BS, which is a very lethal disease for salamanders. Next slide, please. So interestingly, we found even Sicilians being infected by BD. And there are reports of Sicilians are amphibians which are subterranean. They don't have limbs at all, and they live underground in the soil. And we have a very rich diversity of Sicilians in the country. So we also found Sicilians having the infection. So the important thing to note here is that there are many species of amphibians that are getting the infection, but they are clearing it out, or they are surviving with the pathogen for some reason, which needs further investigation. Next slide, please. So we went on to also look at a wild population, an in situ population in Maharashtra. It's called now called Tilari Conservation Reserve, and we identified streams there, and we looked at 2 species primarily, but there are 27 species in those in that area. We focused on 2 species. One is a very common species called the skipper frog, and the other one is called as another a litter frog, which is called as Indirana chiravasi, an endemic frog to India. But Euphlyctus is not endemic; it is a widespread species. So we tracked this population over a period of 4 years now. Can you go to the next slide, please? So these are pictures of the field team investigating the populations in the wild. So on the right side, you have the image of the frog, where we insert a small chip. It's called a RIFT tag or a PIT tag, passive implant transponder. It is just slipped under the skin. It causes no damage to the frog, and it survives. And you can read it with this machine, a reader, and then you can find out whether it was a tagged animal or not. And every time we swab, and then we take weight and the body measurements of the frog. Next slide, please. So this is an animal which is being read out. So you can see that the reader, the animal is placed in plastic bags, and then we read it out. We see if the animal is being captured before, then we release it back into the same spot where we captured it. Go to the next slide, please. So the idea was that it's such a widely distributed frog, and we have no idea of about their local populations and also the status of BD in them. So we followed them over a period of 3 years. We went repeatedly 2 times a year and studied them, repeated the surveys, and marked the newly captured frogs, identified the ones which are recaptured, and tried to find out what was the population size. Go to the next slide, please. So it's again a very busy slide, but I would like you to focus on the left-hand side top graph. So you have 2 lines which are moving up and down. One is the total captures, which is fluctuated from the first year onwards, but after that it remained roughly stable. But if you see the prevalence of BD, that is fluctuating between 80 and 100%. That means almost all the frogs are infected by BD. So this is the development that has happened after the new primers have been used. So we are able to detect BD very effectively in the population. And on the right-hand side graph, you have the total captures and the average infection load. Now, this is very typical of a situation which is globally referred to as cold spot of infection. That is, the prevalence is high, but the loads are not high. Average load is less than 100. So, which is a good sign in the sense there are some strategies in these animals which are allowing them to overcome the infection. Now, that also gives us insight that the infection, the pathogen, and the host are developing some strategies. One-upmanship, like a war that is happening with the frog and the pathogen. That the frogs are trying to overcome the infection, just like the way we have overcome the COVID. The frogs have also overcome the BD infection. And for the first time, the tables that are on the lower half of the slide, don't worry about the numbers, but I just wanted to put them because these are the first estimates of the population of any amphibian in the country. And I think it is very important to do this. I'm sure we will get better and better at it if we attempt and if we keep doing it. And there are many young people who are extremely interested in such studies. And I hope that this will inspire them to take forward such work in the future. Next slide, please. So that's all I have. I just wanted to introduce a new book that was released by the Central Zoo Authority, which many people have contributed. I put the QR code. Please scan it and go and access this PDF of the book. And it talks about management of amphibians ex situ. And we have included sections on frogs, salamanders, and caecilians. And very thoroughly compiled information for any avid amphibian enthusiast. And I would like to look at it and also give us feedback if you have any suggestions to make. And the list I have for people whom I should thank is long. The Central Zoo Authority has been a constant source of support for the lab where I work, and they have guided us, they have supported it financially, which I'm very thankful for. And Wildlife Institute of India was my earlier place of work, and they supported the field surveys for the Himalayan salamander, which I continued after I came to this new institution called Center for Cellular Molecular Biology. Then CSIR, which is a parent body of CCMB, is very generous with its support for such work, so I'm able to continue this. Padmaja Naidu Himalayan Zoological Park is a wonderful zoo, one of the proactive and very well endowed zoo in terms of its location, funding, support that it receives both internationally and nationally. And the West Bengal State Forest Department is also very encouraging with the support with the crocodile salamander breeding project. The Maharashtra State Forest Department supported our work in Tilari. All these people, Sarbani, Barka, Shruti, Suresh Kumar was my colleague at the Wildlife Institute of India. Kaushik Dyuti wrote a book on Himalayan salamanders. He works at the ZSI. He is also a very important colleague for me to discuss many issues. Milind, Gayatri, Harika, Yashwant, all of them worked on the BD pathogen. And they worked in designing primers, testing, culturing the pathogen, and also doing the field surveys. Thank you all. We can take questions.
Thank you so much, Dr. Karthik, for giving a comprehensive overview of the species and the research that's being done on them. So we'll move on to question answers for today's session. So the first question for you is, how suitable is the consideration of XC2 of merging in situ and XC2 efforts, considering the spread of Batrachocytrium?
Okay. Uh, See that it's true that we should be careful when we keep animals both in situ and ex situ. I think the precautions at the level of ex situ is much higher because we are keeping them under our care. Therefore, we should make sure that they are disease free. Now, given the situation that we are trying to understand in the wild, it looks like the disease is not very serious in amphibians in India, but certainly it needs to be looked at very carefully. No, maybe it affects breeding. We don't know. So we have to be careful and follow it up. As many tests as the zoos can try and do, it's a very simple test. Just swab the animal and send the swabs to us, and we can tell what is happening. So I would encourage the CZA to ask zoos who are interested in keeping amphibians in captivity to uh, do more of this testing.
Right, sir. And sir, the next question for you is that you mentioned that the conservation efforts need to be strengthened in the range of the species. Are any locations suitable for species reintroduction? Like as if now it is only in one location in Padmaja. So are there any other areas that are suitable for reintroduction of the species?
No, for reintroduction or for more ex situ populations?
Both.
Okay. For reintroduction, there are some suitable sites, but those wetlands also need restoration and protection. Some in situ conditions have to be brought in, and then we can reintroduce them. For your second part, which is for the ex situ populations, already Gangtok Zoo is identified as a participating zoo. So I don't know whether Gangtok Zoo has any population. I don't think so. But surely there should be a more coordination. And even Manipur Zoo is one of the participating zoos. But I wouldn't recommend moving the animals from Darjeeling to Manipur because I told you that there could be a completely different species.
Right, sir. And so the last question that is there for you is that are there any citizen science initiatives for amphibians in India, like the North American Amphibian Monitoring Program or the Frog Watch USA?
Yeah, there is one, and there is a very active one in the Western Ghats, which Dr. Guru Raja is uh, is carrying out, and it is on one species which is called as the Malabar uh, tree toad, and they are doing a citizen science supported monitoring of the population, more distribution, mapping the distribution of the species. But I would like to emphasize more on um, the stream amphibians and the endemic ones, and they are probably going to be very important if we were to monitor in situ populations. So even the mark recapture study that we are doing, we are trying to get more volunteers involved and get them engaged in trying to learn about these techniques and also the problems that these amphibians face.
Okay, thank you, sir. With this, we come to the end of our today's day 2 of the week 75 Know Your Species talk on the Himalayan salamander and other alone amphibian species. So thank you so much, Dr. Karthik, for taking time out of your schedule and joining us for this concluding week Amrit Mahotsav talks. And I would also like to thank the audience who have been with us throughout. And I hope you know more about the Himalayan newt and the conservation and the research that's being done on the species and the conservation efforts that are going in towards saving the amphibian species. And I would also like to inform them that we'll be back with our concluding talk for the 75th week Azadi Ka Amrit Mahotsav Conservation to Coexistence campaign. So do tune in for tomorrow for our 75th Know Your Species, Know Your Zoo talk on the Great Indian Bustard and the National Zoological Park, Delhi. So at 4 p.m. tomorrow. So do tune in for that. Once again, thank you so much for everyone to joining. Namaskar.
Thank you.